Hello, everyone. Thank you for the invitation to give this lecture. Do not read this slide. So, as an introduction, I said I have set up a few general slides so that you understand the complexity of the issue we are talking about. So the first question is, how is a tumor diagnosed? And on this slide, I show you a summary of the NCCN guidelines for a selected tumor. So when you look, there is a physical examination, medical history, laboratory test, imaging, all kinds of data. And once there is like a hint that there might be a possible tumor, then there is a biopsy, there are additional tests. And once we have all these tests, then there is treatment. Also, don't read this slide. Okay, this is just for your overview. This is a selected example for one tumor. So let's say there is a disease, and according to the guidelines, there are different steps based on what the exact diagnosis was. So like what the spread of the tumor is, whether there are possible markers. And at one point, there is a treatment which we call a systemic therapy. This is like a drug treatment. So this is easy. Okay, let's say that we have all this pipeline for a breast cancer patient, and the pipeline said that we need a systemic therapy. Do not care about the others now. Okay, but what should we give to the patient? Again, I show you the example from breast cancer. So there are two types of drug, those which can be given in monotherapy. So this means that we just give this drug, and there are also combinations. So do you have any idea how these drugs are selected for a given patient? It's based on the preference of the oncologist today, because there are no tests to select the best treatment for a patient. So this is, in other words, a very complex issue. But not only this one is complex. When we look on the structure of the cells, how we understand today how the cell works, this is an image of the EGFR receptor, which is a key receptor in cancer treatment. So here we have the receptor itself. But it's embedded in the cell with lots of other structures. So we have something which is very, very complex. So what could we do? And now we move on to our study. So what we do is we develop software for filtering targets, therapy targets. So what we do is that first we collect data, clinical and transcriptomic data from patients. This means that the tumor is removed. From the tumor, all the genes are measured, and we also save the medical history of the patients. So we know the treatment and we know the follow-up, how long the patient survived. Then we make a bioinformatic tool where we analyze this data. I will show you an example for this. We filter for druggability, and at the end, we find those biomarkers and therapy targets which can be used in clinic to treat a patient. We developed several such tools. So these are some of our bioinformatic tools which we developed in my lab with my colleagues in the last 10 years. And I will show you one example where we used the KM plotter and the rock plotter. Because of the time constraints, I do not show results from the other ones. And to select something, I've selected one example for demonstration, and this is immunotherapy. So immunotherapy is a recent development in cancer treatment. It's a new type of drugs where we activate the immune system. Why do we need to activate the immune system in cancer? So there is the periphery. Periphery is where the tumor itself is. So there we have a tumor cell. And we have the lymph node. The lymph node can be imagined as like a central hub for the immune system. And from the lymph node, we get the T cell. The T cell's job is to destroy the tumor cell. However, this is not always possible. So there are like ligand receptor combinations, like the PD-L1, PD-1, by which the tumor cell can stop the T cell. So it's like a pass on the, this is why I have this image, it's like a pass for the subway system. So the tumor cell has this pass, so this means that the controller will let the passenger or the tumor cell go on unnoticed in the body. So in other words, if we can stop this interaction, we can take away the pest from the tumor cell, and we can activate the immune system. And exactly for this, I've selected one study where such a treatment was developed, and this was published 2 years ago. And this was for rectal cancer. Rectal cancer is a very frequent cancer, especially in males. And the standard treatment for a patient is chemotherapy, radiotherapy, and surgery. There is a subgroup of patients who are mismatch repair deficient. And this is about 5 to 7% of the patients. And this group of patients, they have extremely bad prognosis. And the reason of this is that these tumors are highly aggressive and they do not respond to chemotherapy. However, because this is mismatch repair, this means that there will genetic damage will accumulate in these tumor cells. When there is genetic damage, this means that there will be lots of altered proteins which the immune system could recognize. So the idea was for the authors that, okay, let's try to give an immune checkpoint inhibitor treatment for these patients. And for this, they selected the patients. They selected for the patients so that they looked for the mismatch repair deficiency by checking the presence of 4 genes by IHC. And then they gave the treatment. So they selected the patients. This was a clinical trial. And then they gave an anti-PD-1 monoclonal antibody, which was called dostarlimab, but for our sake, the drug is not important. So they gave the drug every 3 weeks, 500 milligram in 9 cycles. And the plan in this clinical trial was to check the progress within 12 months. And the concept was that if there is remaining tumor or there is progression, the patients will receive the normal treatment. As I said, the normal treatment is radiotherapy, chemotherapy, and surgery. And if there is complete response, so this means that if the tumor is gone, then the patients will not receive chemotherapy, surgery, or radiotherapy. And this is just an example about 2 cases. They had altogether 12 cases which they studied. So this is the initial phase. This is the baseline at the beginning of the treatment for both patients. And this is endoscopy. So this is an image. This is an MRI scan. Here we see the tumor, and this is a PET scan. Here we see the tumor. This is the uterus, the bladder. So this is the tumor itself. And after 3 months, for each of the patients, the tumor was gone. So you see there is no tumor here, and this remained also after 6 months. Also in the other patient, the tumor was completely gone, also after 6 months. There was a follow-up, also after 12 months, the tumor was completely gone. This was the first such study in medical history where a drug was able to cure a cancer. So we can say that this is was a breakthrough. The drug was the drug got accelerated approval. Okay, so what do we do with this study? So let's go back our project so that we take the cancer patient, we get the gene expression from the tumor, and we get the treatment and outcome data for these patients. And then we integrate this into a bioinformatics tool where we can analyze all this data. So what we made for immunotherapy-treated patients, we established a similar database. So we looked up large public repositories about studies where patients in clinical trials were treated with immunotherapy inhibitors. So we checked about 500 studies. And this is a filtering steps, what we made. It's the simple single steps are not important because the brevity of time. It's important that at the end, we had 1,500 patients. And for the 1,500 patients, we have all the data. And of this 1,500, we have about 800 which were treated with PD-1 inhibitors. If you recall here I said that they've checked these genes to select the patients. So to demonstrate what we do, we recheck these genes also in all patients. And as you see, this is MLH1, MSH6, PMS2. Each of these genes reached a high statistical significance in discriminating those patients who respond to this treatment from those who do not respond. So in other words, using our bioinformatic approach, we were able to predict the result of this study, which was actually successful. However, we also have some additional results. First of all, these genes were not the best genes. There were 34 stronger genes predicting response. This means that we can use 34 additional biomarkers to select patients who are eligible for PD-1 inhibition therapy. And another result is that in our database, we do not have only rectal cancer patients, but we have different kinds of tumors like melanomas, breast cancer, and other types. So because these genes are working in other tumors, this means that the treatment can also be given to other patients as well. So at the end, let me give you a very short summary of what our work is about. So we develop software to improve oncology. And how do we improve? We make a bioinformatic test of all genes which are available. And our goal is to choose the ones which are promising candidates as biomarkers or as drug targets. And at the same time, we want to eliminate those markers or genes which are most likely to fail and are, of course, not reliable for patient treatment as well. So with this, I'd like to conclude, and thank you for your attention.
Thank you very much. If you have a question to Professor Győrffy, please raise your hand. If not, then thank you very much again, and I